we came to understand that variation in genotypes appears through mutation, and we explained that this variation would allow appearance of proteins with different functions, and these proteins can account therefore for different phenotypes appearing. And today I'm going to focus on recombination and explain what recombination is and how it allows for new genetic information to appear, and through a specific example explain that variation in the genotype due to recombination leads to change and variation in the phenotype. Recombination is a powerful mechanism that generates variation of genotypes. Recombination takes place during a very special event, which is the formation of gametes, i.e. sperm and egg cells. This process of formation of gametes is called meiosis, and the recombination event takes place through a mechanism called crossing over. I'm going to explain these terms in the coming slides. Crossing over is a swapping of genetic material that happens during the formation of sperm and egg, i.e. during meiosis, when paired homologous chromosomes align and exchange segments of their DNAs. Let's first see the case of a meiosis happening without recombination first. We are familiar now with these depictions where we can see two homologous chromosomes, i.e. chromosomes that bear the same genes in them. They can have different alleles maybe for the same genes, and this one was inherited from one parent, this other chromosome is inherited from another parent, and in this representation, these homologous chromosomes were actually duplicated. Each one of them is made of two sister chromatids that are still attached to each other. So these replicated homologous chromosomes, they can undergo meiosis, whereby each of the chromatids will get separated and will end up individually present in one of the gametes. In this case, I'm representing sperm cells. It could be also eggs. And you can see we start with these 4 chromatids, and each one of them is inherited in one of the sperm, which then would go and fertilize an egg and bring half of the genetic information. But the point here is that without any exchange happening, these two genes, as you see them here, the green and the red gene, which could have different alleles in the yellow chromosome, the homologous chromosome, these two genes will always remain together. Green with red remain here, they remain here. The light green with the light red also remain together, and we end up with only 2 types of gametes generated in this case without recombination. Now we will look at the case where there is meiosis involving recombination. And in this case, the 2 homologous chromosomes which form this pair, they will remain together. Not only that, they will overlap 2 of their chromatids, one on top of the other. And this crossing over of one chromatid over the other one is what allows the recombination event to take place. So recombination meaning DNA will be broken and then stitch it together. And what you see happening here is at the level of the crossover, there is a breakage of both chromatids, and then the crossing over will allow the exchange to happen. And when the DNAs are restitched, relinked together, this blue chromatid now of this chromosome has in it part of the yellow chromatid, and the yellow chromatid had gotten a blue segment of the other chromosome. So this exchange that happens would allow, therefore, the formation of different types of gametes, because following the same process, when meiosis takes place here, the 4 chromatids will get separated from each other. However, in this case, we get, in addition to the 2 types of chromatids that we have seen in the previous scenario, we have 2 new other chromatids that are generated through crossing over. And in these chromatids, the genetic information is no longer the same. It's not the same because this allele of this gene here now is in the same chromosome with another allele that it did not used to be with. And the gametes that we will end up having will be different, therefore, and they will have a new type of genotype in them compared to the previous scenario. So this explains then that when crossing over happens during meiosis, it generates genetic variation by mixing together genetic information coming from two homologous chromosomes and generating new genotypes in these gametes. So crossing over results then in a shuffling of genetic material and generates gametes with the new genetic variation that produce offspring with variant traits. So what I'm trying to say here is these gametes now, because they have a different genotype compared to other ones, and their genotype is new. It did not exist before. It only existed due to this recombination event and crossing over, now can fuse with another gamete, an egg, for example, and because it's carrying a new variation of the genotype, it can lead to new traits in the offspring. So let's take this example where the color code of mice has been studied, and we will see that the recombination events that take place allow the generation of new traits in the mice population. So let's consider the original parents, one with dark color coat, the other one with a really light color coat, and imagining that these are coming from pure breeds, so they have the same type of alleles in their homologous chromosomes, and when they mate, they generate the first generation called F1, which inherits half of the genetic information from the dark-coated parent and the other half from the light-coated parent, and its phenotype is intermediate, as you see, between the light and the dark. Now, considering F1, if we cross F1 mice amongst themselves, the crossing over events that take place during the meiosis in the male and the female will generate different types of gametes where the genetic information will be shuffled, which means that the genes that control this trait will be also shuffled. And this leads in the second generation called F2 to this continuum of expression of the trait here being the color of the coat, having two extremes, either the light or the dark, and we have other expressions in between. So you can see that these mice, when we look at their genotype, each one of them has a unique genotype that corresponds to the trait that is being expressed. Where, for example, this one has most of its genotype coming from the white genotype, and this one has most of genotype coming from the black mice or the black chromosomes. The ones that are in between, they have different ratios of the alleles that they gotten either from more of the black or more of the white.